Hi, good evening. I'm Dr. Philip McMillan. Thank you again for joining me as we continue this journey of exploration around the COVID 19 pandemic and some of the unanticipated outcomes that would happen in it. And this is a very interesting thing, and I'm going to be sharing with you a paper. And this paper was only published today about the methyl pseudouridination of mRNA and ribosomal frame shifting, a complex topic. And I've decided to take it on because I think it is actually important. So, here is the problem that we've got. Whenever you develop any kind of new technology, you're going to be going through a period of trying to anticipate and work out some of the things that you never thought could happen that happens. These are the unknown unknowns. And in the context of the COVID 19 vaccines and the mRNA technology, even though this had been tested before, it has never been tested at scale and at speed. And so I think for anyone to presume that they aren't going to be unknown unknowns is a bit foolish and naive. We give credit for the fact that they were able to do something so quickly. That was able to have immune responses, but that's a separate focus from my perspective where I look at autoimmunity. Anything about stimulating the immune system, I get worried about. But that's aside. It was quite a remarkable achievement to bring this technology at scale. But in truth, you had to experiment on the population. And it's not clear that people understand that clearly enough. So here we are now coming into the area of unknown unknowns. That's what I mean. So, in order to be able to do this, I'm taking on trying to explain some of the complexities around how cells work. So you have to bear with me. And even the ideas that are shared in this are quite complex. I really spent most of the day reflecting on it, trying to think about how I could explain it. So if I don't get everything right, please be patient. So, we're going to start with some basic things about what they were doing when they were coming up with the idea of the vaccine. So, you have to realize, so this is talking about, this is what the Nobel Prize was about, the pseudouridine in mRNA COVID vaccines. So, this was a paper that was produced in 2021 here. And what they were looking at is they were talking about how important these pseudouridines were to the mRNA COVID vaccines. And first, understanding that and understanding what changes they made with regards to it is absolutely critical. So they had a few problems with the mRNA technology when Malone had come up with the idea. The problem was that the immune system recognized it and destroyed it. So if you were going to try and get it into the cell to function, how do you get it past the immune system? And so this is where that critical breakthrough occurred when this is correct, and the BioNTech came up with the pseudouridine principle to be able to bypass the immune system. And essentially, this is what it looks like. And this is what the paper shows. This is from the same paper. You have here uridine that looks like this, and then you have pseudouridine. And this is what the research was using instead of actually uracil, which is part of RNA. Uridine can work as well, but pseudouridine seemed to work to be able to bypass the immune system. What a lot of people were not aware of is that in reality, the mRNA or the modified RNA vaccines did not use pseudouridine. It actually used N1 methyl pseudouridine because they had found that this one was even better at evading the immune system. So, although the Nobel Prize was given for this, this was not actually used in the vaccines. It was actually this. Now, it raises a question because I think they did do this with the Zika virus vaccines, and this is what the paper had spoken about. And you can look at the paper and as the history went along. So they did trial this methyl pseudouridine with Zika vaccines, but this was not at big scale. So there would inevitably be some things that you just didn't even know could happen. And this is what I'm going to be talking about here with regards to ribosomal frame shifting. This is a fascinating topic. As I said, I've never heard about it before. And this paper here, it was received on the 25th of January, 2023, was finally accepted on the 31st of October, 2023, and it's published the 6th of December, 2023. So that's just yesterday. The lag time between this could be for a number of reasons. Maybe the reviewers weren't happy about something, they needed things to be changed, but this is a long time to wait because the information in this paper is actually very important and to a certain degree very, very concerning. And so this is a big deal. So, what I'm going to do before I go into that is that I'm going to take on the challenge of trying to explain what this is about. And so, we'll have to take you to just a few simple slides so that you understand what we're talking about here. So, this here is an overview of an animal cell. Okay, so you have this is the whole cell here. And in the center here, well, not in the center, but at the top here, this is the nucleus. That's where the DNA is. And the DNA will then convert into RNA. The RNA will go outside the nucleus, and then it will be converted in this, what we call rough endoplasmic reticulum, into proteins. And you can see here, these little dots, these are ribosomes, and I'll show you what these are. But this is the machinery the cell uses to convert information from the nucleus, that was DNA, which turned into RNA to make it into protein. And so this is the process that happens in this part of the cell. And every cell has this to different degrees depending on how much protein they are making. So this is the part that we're interested in, these ribosomes which do the work. So once you understand exactly what they're doing, you'll see what was the trick that they came up with to work around the immune system. So this is just a picture of a ribosome. And what you have here is this is the mRNA. This is normally what happens. The mRNA is produced from the DNA and it is then sent out of the nucleus. And then these ribosomes pick it up and they almost like a zipper run along it and out comes a protein. And they have these binding sites within the ribosome which then causes the amino acids to dock and connect them together. So it would look something like this, where you have here, this is the mRNA. Let me make this full screen. So again, this is an mRNA starting at the 5' end. This is the ribosome. It would start here. And as it goes along this is showing it going along step by step. These are not multiple ribosomes. It's just the same ribosome going along. And as it goes along this zipper, it starts to produce the peptide chain. And it will look like this. And gradually at the end, when it's finished going through the whole thing, you end up with a protein. And that protein is then processed. So this is what they were using with regards to the technology to make the spike protein. They would then use that same mRNA instead, and well, they'd use the same mRNA or a similar one that didn't need to go into the nucleus, take advantage of the ribosomes to make a protein. And in that context, that was the spike protein. So this is now where it gets to a little bit more complexity. The way how it then works is that you have, this is now an example of an RNA, and it only uses 4 nucleosides here. This one here is uracil, guanine, cytosine, adenine. So it uses just a 4 combination to make any protein that it wants, and it's always in a 3 connection. So 3 of these make 1 amino acid. And that amino acid is then connected, and you have this line of protein that is then formed. And when all of it is formed, it then tends to fold up and then it's processed into the final protein. The critical thing you have to notice is that it's 3 at a time as you move along. Okay. And this is how it works. And actually, you can see it here from the principle of DNA. This is DNA. This would be in the nucleus. And again, this is the double-stranded DNA. The DNA stretch makes an mRNA, which then goes out of the nucleus, and then it goes to the ribosomes. And you can see in this context here, these three, UAC makes tyrosine, then this UCA makes another amino acid. This one here is GGU, makes another amino acid. And then this one here, UCA, makes another amino acid. And this is just how the process works. And it's this that then becomes the protein. So that's the principle of how the ribosomes work. And this is why we now take it back to this frame shifting, what I consider to be the unknown unknown that occurred with regards to the mRNA, specifically, it seems, from the spike protein from mRNA from Pfizer. Now, remember what I said about these amino acids. I think I probably need to get this back. And if you get this, you'll understand why this becomes a problem. So here we have, again, that principle of this mRNA. What they found happened, and you just have to imagine that this U is taken out. And this is what they mean by a slip. Almost, it does a jump. Instead of it going nice, these three, then these three, then these three, then these three. What happens, for some reason they don't know, it misses this. And you can see that instead of UAC, it then becomes ACU, CAG, GUU, CA, and then something else. This is a completely different protein. So just by missing one of these here, you end up with abnormal proteins. And this is something that they've just technically realized, or I shouldn't say they realized it, they've just published about it, which is something. So therefore, this person was researching it for some time, and it's now finally published. And so here is the paper, and I think that this is now the bit that I want to focus on. I've just highlighted a few points around it, and I'll come back to them in a little bit here. What they found was that when the ribosomes made some of these abnormal proteins, they were looking at the interferon response, and they found a significantly higher interferon response to the one frame shifted antigen in the BNT162b2 vaccine group compared to the ChAdOx. This is the AstraZeneca versus the Pfizer. And you can see here, so this is the image that is showing this response. And you can see that when it came to the interferon response to these abnormal shifted peptides, the primary abnormal response was with the Pfizer vaccine. Interestingly, it did not happen with Moderna. We don't know why as yet. It didn't really happen with AstraZeneca, and certainly didn't happen in the untreated case. But this triggered an abnormal immune response, an interferon response, with regards to a number of these abnormal proteins that occurred. This is really, really important because it could start to explain some of the really strange presentations that we have been seeing around what happens. I'm going to show you again so that you get this, how important this question is. Here again, I've got this protein translation. So in this one here, again, you can see how three of them connect to make one amino acid here. So this one here is the UAG. This one here, GUC, and they tend to they balance each other out. A connects with U, G connects with C. So you can see here that this is going along fine and it's producing the protein that it would expect. But if you imagine that suddenly the next one to go along is supposed to be CUA, but it slips on this one and frame shifts so that instead of CUA, you get CAC. The next one down suddenly becomes UAG. UAA, CAC, GUG. This is a completely different protein. Just by that one shift right here, it's because the methods are so precise that even the slightest shift can create a problem. Now, we don't know if this is related to the N1 methyl, as I said, pseudouridine here. So, as I said, it's unclear if this is connected to the fact that they use this as opposed to this. All that research needs to be done to try and see if we can understand, one, why is it happening primarily with Pfizer and not with Moderna, and what implications, if any. Now, some people will be quite happy to say, come on, there's nothing that's happening. I don't really think like that. That's not how science works. This is an unknown unknown. It's triggering an interferon response around the fact that we're getting abnormal proteins being formed. There's still a lot of work that has to be done to try and understand the implications. And who knows what that means? Because sometimes these proteins, if you don't end up, if they can't then make a logical protein, sometimes they get stuck. And then this protein will be destroyed or sometimes get stuck within the cell. Does that explain why we have these strange patterns where spike protein or fragments of the spike protein continue to circulate for long periods of time after someone has been vaccinated? These are very big questions. We don't know what it means, but it definitely is unexpected. This is the reality of science. And this is what oftentimes happens with many products. There are many drugs that were in production, they were doing well in the phase one, the phase two, then they hit phase three or even phase four, and you see a pattern that you didn't anticipate, and this is a problem. We don't know if this is one of them, but all we know is that it was unexpected. What else don't we know that could be occurring? These are the challenges that are ahead of us. And from a clinical point of view, it makes it extremely difficult for us to know exactly what kinds of diseases we can see presenting, if any at all. That's always the hope, that it means nothing. But if it's triggering an interferon response, I suspect that's not going to be the case. Let's hope that these things are mild. But there is still a lot more research to be done in order to have a full understanding of longer term implications. Have a great evening.